Hello, everyone. I'm Shannon Wyman, Director of Program Development at Keystone Symposia, and it's my pleasure to welcome you to today's ePanel. Today, we will be featuring the awardees of the Michelson Philanthropies and Science Prize for Immunology, which aims to support transformative research in human immunology to accelerate vaccine and immunotherapeutic discovery. The prize is a collaboration between Michelson Philanthropies and Science AAAS to award $30,000 to young investigators advancing immunology and immunotherapy research and applications. Here to share a few remarks about the prize is Alya Michelson, co-chair of Michelson Philanthropies.
Hey everyone! It's a pleasure to be here with you today to celebrate the incredible scientist recognized with 2024 Michelson Philanthropies and Science Prize for Immunology. This is my first time addressing the participants, and I'm truly excited to be part of this moment. The work of these brilliant minds advancing our understanding of the immune system, pioneering new vaccine approaches, and pushing the boundaries of cancer immunotherapy holds immense promise for the future of medicine. Their creativity and determination are driving breakthrough that will improve and save lives. With my husband, Dr. Gary Michelson, I want to express our deepest gratitude to Holden Thorp, Bill Moran, and their team at Science. Your dedication has been vital in bringing this program to life and ensuring these young scientists get the recognition they deserve. I'm incredibly proud that our prizes have recognized more female luminaries among young researchers than any other. As someone with an immigrant background, I find it very important to see such demographic and ethnic diversity. It's crucial to create opportunities for all talented minds to shine. By investing in their work, we advance in science and building a healthier, more equitable future. I'm also thrilled about another significant milestone this year, the launch of the California Immunology Institute, the first of its kind worldwide. This new hub will be dedicated to nurturing the most promising ideas and future star projects in immunology, some of which, as we hope, will come from our Michelson Prizes. We're excited to see what groundbreaking work will come from there. A big thank you to Keystone Symposia for organizing this program, and congratulations to this year's recipients. We are so proud of all you've achieved and look forward to seeing the incredible impact of your work. Thank you.
Wonderful. And now we have a few words about the prize from the team at Science. First, we have Dr. Holden Thorp, Editor in Chief.
Hello, everyone. It's great to be with you. I'm really excited about this year's winners and this prize. First, I just want to thank the Michelsons and the Michelson Foundation for their support and for the support that they have for the way we do these prizes, which is to identify researchers early in their career who can contribute to, in this case, immunology for many years to come. It is very, very challenging. And I write about this a lot if you follow me, to build a career as a PI studying science in academia. And it is really important for us to provide encouragement to the people who are willing to take that on, because we need a great supply of new scientists leading us into the future. And this year's winners are certainly all examples of that. I want to thank my colleague Sarah Ross, who you're going to hear from in a minute, for providing the disciplinary expertise that is needed to do this kind of thing. It is a lot of work to read all the essays and interact with the other folks who are helping us make these determinations. And Sarah is a terrific colleague that we are very appreciative of, and she did a great job here identifying today's winners. I'm really excited to listen to the talks. Now I want to turn it over to my colleague Bill Moran, who is the publisher of the Science family of journals and is responsible for a lot of the work that goes on in establishing these prizes and making them the success that they are. So now turning it over to the publisher of the Science family of journals, Bill Moran.
Thank you, Holden, for that lovely introduction. My name is Bill Moran, and I am the publisher for Science and the Family of Journals. I want to take a moment here, one, to congratulate the award winners. This is what it's all about. You're going to find out more about the research that won and has been recognized by Sarah Ross and her team. And you'll see the future implications for this in cures and saving lives and how crucial this is. Now, I want to take a moment, though, to talk about why we do this prize. And in particular, I didn't work with Dr. Michelson on this, but if you have a moment, I was looking at the Michelson Philanthropies website the other day, and there's an excellent article from Dr. Michelson, and it talks about the NIH and the good things at the NIH and some of the bad things at the NIH, or I should say, things that need to be improved. Let's be positive. It's always good to be that way. But why we started this prize? We started this prize because Gary points this out, that when PIs are first awarded their grant, the average age is 44. That's not young. That's not early in that case. And this prize was developed to recognize and to help bring attention to early career researchers. And we have accomplished that with the help of Michelson Philanthropies. So this was the real reason for this prize, and the prizes that we do at Science is to really recognize early career researchers. And if you read that article from Gary, you'll see he has some solutions in there. And I did enjoy reading about his suggestions. So I would highly suggest taking a look at that article. And then real quick, I do want to talk about Science for a second and AAAS. Just to give you an overview of who we are. AAAS was started in 1847. So we have a long pedigree. But that pedigree is also Science Without Borders and collaborations with organizations like Gary and Michelson Philanthropies. And it's also our way of giving back to the community, to supporting the scientific enterprise. And then around 1880, Science was invented, was founded. It was from some seed money from Thomas Edison, and I think it was about $10,000 that he put towards Science. And we've evolved a lot since then. But just want to give you some background on Science and AAAS. And before I pass on to Dr. Sarah Ross for the real heart of what this is all about, the research itself, I do want to thank the Michelsons and Michelson Philanthropies. I'd also like to point out thank you to Jeff and Michelle and Roger Gonsalves and Holden Thorp for all of their help with this prize. It wouldn't be successful without the support and recognition of the editorial team actually recognizing those winners. So I'm looking forward to hearing from Dr. Sarah Ross about the research itself. So over to Sarah. Thank you.
Thank you for the kind introduction. So the Michelson Philanthropies and Science Prize for Immunology was established in 2021 to recognise recent cutting-edge work by early career scientists in the field of immunology. As Shannon said, the prize has a focus on recognising transformative research with the potential to impact how we understand human immunology, advance vaccines, or develop immunotherapies. Entries for the 2024 prize were accepted from early May until the beginning of October 2023 and open to individuals with an MD or PhD aged 35 or younger. Entrants were required to submit an essay that was no longer than 1,000 words and that described their contributions to human immunology research within the past 3 years. This year, we continue to have outstanding entries showcasing high quality and innovative research through engaging and compelling essays from many talented young researchers from across the world and who also span different early career stages. The essays highlighted the many different areas of immunology actively being studied, from understanding the role of the immune system in protecting humans from pathogens like SARS-CoV-2 to the contribution of dysfunctional immune cells to diseases like cancer and autoimmunity. Some researchers tackled fundamental processes like how different types of immune cells develop, while other scientists aim to identify biomarkers of disease. Methodologies implemented range from computational approaches to extract information from genome-wide data to careful and comprehensive experiments that elucidated molecular mechanisms underpinning how the immune system functions. After the closing date, each application was assigned to 2 editors from within the science family who independently evaluated each composition. The essays were rated in 2 areas. First, for their scientific quality and significance, and second, for the clarity and style of the writing. Based on the editor ratings during the first round, we shortlisted 8 outstanding applicants who were then assessed by a jury comprising both science editors and leading researchers in the field of immunology. While it was not an easy task to determine the winner and runner-up, 3 entries did stand out. Our first runner-up, Gabriele Casirati, wrote an essay that impressed during both judging rounds for its detailed description of the challenges of cell-based immunotherapies for cancer and his research towards a cutting-edge gene editing strategy that has the potential to help overcome the current limitations. Our second runner-up, Carla Nowosad, composed an essay that editors and judges found to be compelling and which conveyed in a very accessible way elegant fate mapping approaches to understand fundamental questions about the interactions between B cells and the microbiome in the gut. Finally, our winner, Bingxu Liu, submitted an outstanding essay that describes the discovery of the first known immune sensor in human cells to transport ions in response to pathogens. The essay was clear and engaging, and the editors and jury were excited by the fundamental, groundbreaking nature of the work, as such molecular understanding is essential to drive the development of next-generation vaccines and cancer immunotherapies. Now, I'd like to conclude by thanking some of the many individuals who helped ensure the success of this year's prize. Firstly, I'd like to acknowledge editorial coordinator Mary Rose Madrid, who organised judging assignments and helped us keep to our deadlines. I also like to thank Roger Congalves and the Science Prize team for liaising with the Michelson Philanthropies. I'd also like to extend my thanks to colleagues at Science and Science Immunology for evaluating the essays. In particular, I'd like to highlight Seth Scanlon, who was previously the immunology editor at Science and who now leads the editorial team at Science Immunology. And he was really instrumental in coordinating this year's judging, along with the previous rounds of this prize. Thanks also go to members of our grand jury, Dr. Andrea Schmitz at Technical University of Munich, Dr. Philippe Bessot at Institut Pasteur, and Professor Peter Oppenshaw at Imperial College London, who joined Seth and me to determine a winner. Thanks too to Jessie Slater, Science's research and data analyst, who edited the 3 finalist essays. And finally, thanks to Bill, Holden, Dr. Gary Michelson, and Alya Michelson for the opportunity to recognise and support this year's winners, alongside the other awardees from previous years. Thanks to all applicants, and also congratulations to the winners.
Thank you, Sarah, Bill, and Holden. Now it's my pleasure to introduce this year's awardees. After I introduce each awardee, we will show a presentation about their work. Once all of our awardees have presented, we will have a group Q&A discussion. You may submit your questions at any time into the Q&A box and vote on other questions that you would also like to hear answered. Please note if your question is directed to a specific speaker in the text of your question. And now I'd like to welcome our first speaker, finalist prize recipient, Dr. Gabriele Casirati of the Dana-Farber Cancer Institute at Boston Children's Hospital.
Hi everyone, and thank you for the fantastic introduction.
Great. And Gabriele studies how to utilize gene editing of hematopoietic stem cells to enable targeting of hard-to-treat hematologic malignancies like acute myeloid leukemia. While therapeutic antibodies and CAR T cells are able to target these malignant cells, the antigens they target are also present on healthy cells, resulting in off-target toxicity. Gabriele has found a way to introduce single amino acid mutations in these surface antigens on the healthy cells such that they are resistant to the cancer therapy. His presentation is titled Stem Cells in Disguise: How Epitope Editing Can Empower Targeted Cancer Immunotherapies.
Good day, everyone. I'm Gabriele Casirati, a research fellow at Boston Children's Hospital and Dana-Farber Cancer Institute. And today I'm going to present you my project, which is termed Stem Cell in Disguise, or the use of epitope editing to enable a targeted immunotherapy for acute myeloid leukemia. So acute myeloid leukemia is the most common type of acute leukemia in adults and second by frequency in children. And despite the recent introduction of several new agents into the clinical practice, it is still associated with a very poor prognosis, with less than 30% overall capable of achieving long-term survival. And one of the other typical treatment that is typically being employed is allogeneic stem cell transplantation, which is the oldest type of cellular immunotherapy that we use for hematological malignancies. But over 50% of AML patients relapse even after allogeneic stem cell transplantation and are left with very few therapeutic options. And again, even in this case, relapse still remains the main cause of death. So regarding immunotherapies, we've seen tremendous advances in recent years, especially since the introduction of CD19 targeted CAR T cells, which has revolutionized the treatment of these phenotype hematological malignancies. But you may want to apply the same strategy also for acute myeloid leukemia treatment, but unfortunately the choice of targets is very limited because AML cells express on the surface targets that are typically co-expressed also by healthy hematopoietic progenitors or differentiated myeloid cells. So you would end up eliminating both types of cells, and that this would create severely limiting toxicity. And this is what we call on-target off-tumor toxicity. On the other hand, you may also end up creating extra hematopoietic toxicity if the target is also expressed by other organs. But to give you an impression of the problem, I'm sharing here a slide that was presented by Veronique Lisier at the last year ASH annual meeting. And this is an analysis of the Leucigene cohort of single-cell RNA-seq data where the expression of surface markers was compared between acute myeloid leukemia, leukemia stem cells, and healthy hematopoietic cells. And the fact here, you can see there are a number of potential candidates, but the majority of these are still expressed by extra hematopoietic tissues, and the remainder are expressed also by other hematopoietic cells. So again, you have very few targets, or next to none, that are potentially actionable without creating potentially life-threatening toxicity. And this is probably the reason why in 50 years of approved treatment options for acute myeloid leukemia, we've never seen really the introduction of new targeted immunotherapies options, except maybe for the use of CD33 targeted antibody drug conjugates. But so this is the problem. We have pretty much a lack of stringent leukemia-associated antigens that are safely targetable, and because it would end up creating on-target off-tumor toxicity, and this would result in hematopoietic aplasia or severe neutropenia, for instance, that cannot be sustained for long time periods. Again, you may want to limit in time the exposure to your targeted immunotherapy, but this is likely going to reduce your chances for disease eradication. And on the other hand, there have been a few potential solutions to this problem that have been proposed, such as the genetic knockout of the target antigens in hematopoietic stem cells that are used for bone marrow transplantation. And therefore, the new hematopoiesis that would reconstitute in the patient would be resistant to the CAR T cells or your antibody treatment. But even in this case, the problem is that this strategy is only applicable to genes that are irrelevant for hematopoietic development, and therefore they are also more likely to be lost by the leukemia cells, therefore resulting in antigen-negative relapse. So our idea here was to instead employ an approach that we term epitope engineering or epitope editing, which means that we are not going to genetically remove the gene by knockout, but instead we are really reducing the engineering to the bare minimum. So we're just changing one amino acid residue at a time, so that is sufficient to avoid recognition by the targeting antibodies or CAR T cells. And this while preserving surface expression, the regulation, and the function of the gene. And the idea here is that you can therefore target potentially any gene on the surfacium, and therefore genes that are also essential for AML survival or proliferation. again, regardless of their expression by healthy hematopoietic stem cells. And in turn, this would reduce the risk of tumor immune escape. So we started with some candidates, and the first that we worked on was FLT3, a very well-known target for acute myeloid leukemia. It is expressed in the vast majority of cells and by the vast majority of patients. And luckily for us, there were already some targeted immunotherapies in clinical development. So what we did was design a pipeline for variant identification. So we screen libraries in human cell lines that express different mutations in the extracellular domain, and then we FACS sorted these cells based on their negative recognition by the therapeutic antibody. And then we sequenced these cells and then recloned and revalidated this surface antigen variant to make sure that they preserve the functionality of the gene. And in this case, we're talking about a cytokine receptor, so we are mostly interested in its capacity to bind its natural ligand, the FLT3 ligand. So for instance, in this case, you see that in this library region, we identified a single mutation that was sufficient for the abrogation of the therapeutic antibody binding, here in position 399. And then we on to validate that this does indeed remove nearly completely its affinity to the antibody or CAR T cells, while still preserving FLT3 ligand affinity and downstream kinase phosphorylation. And we ran a number of additional validation tests to show that we don't see any difference from the wild type variant. So it seems to be perfectly functional. The other important point was the capacity to introduce this mutation into primary CD34 positive HSPCs. And luckily for us, this mutation was potentially editable through adenine-based editing, which is a recently introduced technology by the lab of David Liu and Nicole Godelli that can efficiently introduce single base changes without the need for DNA double-strand breaks or the need for DNA template donors. And we were able to identify combinations of base editors and guide RNAs that were introducing this mutation with high efficiency. This was true in CD34 positive HSPCs. As you can see here, efficiency can go up to 80 to 90%, and this is the same across different stem cell subsets, including the CD90 plus 45 minus stem-enriched subset, and without any effect on the stem cell surface phenotype and also stem cell functionality. We validated this both by in vitro and in vivo assays. We also, in fact, transplanted these cells into immunodeficient and MBSGW mice and compared this with control edited cells edited in a neutral locus such as AAVS1, and we did not observe any difference in terms of engraftment capacity or multilineage differentiation capacity, both in primary and secondary recipients, and the cells were not counter-selected by looking at the editing efficiency over time. So we can conclude that there is no effect of the editing on the in vivo cells. And this is also especially true because FLT3 ligand is cross-reacting between human and mouse. So we next confirmed that this mutation actually resulting in protection from the CAR T cells against FLT3, and we performed in vitro co-culture killing assays here on the left with human cell lines that were expressed in the FLT3 variants, or human CD34 HSPCs on the right. They were either edited for FLT3 or the control locus to show a difference in survival when cultured with the CARs. But obviously we wanted to move to a more comprehensive evaluation of our proof of concept. So we designed a more complex in vivo setting where MBSGW mice were first humanized with CD34 positive HSPCs, either edited for FLT3 or for the control locus, and then in the same mice we transplanted an AML patient-derived xenograft that was FLT3 positive, and then with this condition with these mice were treated with FLT3 CAR T cells. And as you can see here, we were able to see perfect eradication of the leukemia cells when the mice were treated with FLT3 CARs. But again, more importantly for the purpose of our research, we were able to confirm also the resistance of the epitope-edited hematopoiesis to FLT3 CARs. You can see here that we have on the left the granule monoprogenitors, on the right the lymphoid prime multipotent progenitors, which are known to be FLT3 positive. And here you can see in the ABS1 edited condition that are completely eliminated when the mice are treated with the CARs, while when we have the epitoperitic condition, these lineages are preserved, and therefore they are not eliminated, and we can conclude that therefore we are not incurring into that on target of tumor toxicity. But obviously, we also want to make sure that this is not just an isolated case and we can edit more than one target. And so we started the possibility to introduce more than one epitope modification at a time. And this is relevant because, again, AML is very plastic, it's very heterogeneous, and we still have the risk of potentially antigen downregulation and therefore antigen negative relapses. And so we started a second surface target, which is CD123, another very well-known target for AML. It's the alpha chain of the IL-3 receptor. And here we repeated a screen with different base editing guide RNAs, and we identified combination that were able to introduce a mutation that abrogated the binding of a therapeutic antibody clone without affecting the surface expression. And again, we then moved to our in vivo setting, and we transplanted these mice instead with double-edited HSPCs, so both C123 and FLT3 base-edited cells were again the control locus, and then we co-grafted in the same mice an ML patient-derived xenograft that had shown in the past to be partially resistant to FLT3 targeting alone. And in this case, by applying a dual antigen immunotherapy with CAR T cells against FLT3 and CD123, we were able to eradicate this disease. But again, looking at the healthy hematopoietic lineages in the bone marrow at the sacrifice, we were able to conclude that they are protected against the activity of both CAR T cells, because again, you see here that the absolute count of them are comparable to the non-treated conditions across both myeloid and lymphoid subsets. So, in summary, what I hope I was able to show you is that epitope editing confers resistance to CAR T cells or therapeutic antibodies while generating effectively an artificial leukemia-restricted antigen. And epitope engineering can definitely be achieved by adding base editing with high efficiency and minimal toxicity. And again, this editing technology does preserve the stem cell repopulation capacity and the differentiation capacity of the stem cells when the mutation that is inserted has been fully validated to be overlapping with the wild-type gene. And again, finally, multiplex epitope editing is possible and can enable the use of multi-target anti-AML immunotherapies with tolerable toxicity. So in conclusion, I want to acknowledge the contribution to this work. I want to thank all the people in the lab, especially Pietro, all the other fantastic scientists that have contributed to this work, our collaborator, all our funding agencies, and all of you for your attention. Thank you.
Great. Thank you, Gabriele. You can enter your questions for Gabriele in the Q&A box now, and we will get to those when we open up to the Q&A discussion after the other presentations. Our next awardee today is finalist prize recipient, Dr. Carla Nowosad of Langone Grossman School of Medicine at New York University.
Morning, Shannon. Thank you for the introduction.
Wonderful. Carla studies how B cells influence intestinal immunity and microbiome composition. By coupling fate mapping, model microbiomes, and IG sequencing, she has shown that gut germinal centers select B cell clones with increasing affinity to members of the microbiome. Mapping how antibody responses are generated in the absence of infection has major implications for understanding proper control of the microbiome, which impacts many disease outcomes. Her presentation is titled, Who Goes There? B Cells Assess Risk in the Intestine.
Hello, my name is Carla Nowosad. I'm an assistant professor at New York University, and it's my pleasure to be here and be able to tell you a bit about our work, which was featured in this year's Michelson Science Prize for Immunology, about how B cells are able to tell friend from foe in the intestine. My lab studies the B cell receptor, which is a carbon copy of secreted antibody. Antibodies exist in many flavors and play several essential roles in immune protection. There are the natural antibodies, which bind broadly to pathogens early in immune responses, antibodies which recruit other cells and pathways to the site of inflammation, antibodies which bind tightly to pathogens and clear them, and antibodies that regulate the microbiome. Recently, we have become very interested in these border control antibodies that regulate the microbiome, since shifts in the composition of the microbiome can impact the onset and progression of many diseases, including inflammatory bowel disease, obesity, cancer, and depression. Therefore, we think understanding how antibodies regulate this process is essential to try and intervene in the onset and progression of some of these diseases. Antibody responses evolve in these cellular training camps known as germinal centers. Here, activated B cells enter, make mutations to their surface B cell receptor, and become selected if the mutations that they acquired improve their binding to antigen. Over time, B cells with ever-improving binding of their B cell receptors are selected and expanded over and over, so much so that a single successful cell can take over an entire GC, making it what we call monoclonal. Now, we have a great tool to watch this process happen in real time through use of the confetti allele. Here, cells are marked with 1 of 10 fluorescent colors, and if a cell is successfully selected and expanded in the germinal center, it appears single-colored. And here you can see a blue cell that's been very successful in the germinal center and taken it over. Now, we decided to approach this project by asking, can germinal center selection take place in the intestine? Now, the intestine is a very unique environment for B cells, and here germinal centers form even in the absence of infection and are present at all times in 2 locations, the Peyer's patch and the mesenteric lymph nodes. Now, we know for vaccines and infections that germinal centers perform their normal functions, but in the steady state, when there's no infection, things were a bit different. So firstly, these germinal centers have to respond to a massive diversity of antigen. These are antigens that come from the food that we're eating and the microbiome that we live alongside. Other studies showed that non-specific cells were able to participate in these steady-state germinal centers, calling into question whether they were specific for antigen. Other studies found that the cells that produce antibody in the underlying lamina propria frequently were polyreactive. That is that they bound to more than one thing. And this goes against some of the processes of selection towards one single antigen. Couple this with the fact that the intestine is generally considered a tolerogenic environment and that the Peyer's patch plays a generative role in other species to increase diversity, this question was very much still open. So in order to observe germinal center selection in the steady-state intestine, we used multicolor fate mapping using the confetti allele that I showed you earlier. But this time, we made a mouse where the germinal centers were uncolored until we gave a drug known as tamoxifen, and then we can color these cells and watch them select over a specific time period. And here I'm showing you 3 Peyer's patches alongside one another at different time points after turning on the confetti allele. And you can see here our follicles, which are these beautiful, round, colorful structures. And we can see over time that the 10 colors that we see in the beginning converge over time to one color. So we can see at day 21, this beautiful aqua-colored follicle next door to multicolored neighbors. We can also watch this happening in mesenteric lymph nodes. And here I'm showing you a mesenteric lymph node 21 days after coloring with confetti. And I'm showing you in these enlarged windows 2 germinal centers that underwent very different fates. One here became completely monoclonal and is blue, and its neighbor remained mixed. Now we can do this again and again in many germinal centers in many mice, and we can identify these rare bursting events. So these are events where the GC gets overtaken by one very successful clone. And this led us to ask the question, what is so special about being a winner clone? What do winner clones bind to and do in the intestine? The strength of the confetti system is that we can isolate single germinal centers and map their clonal evolution. Here, we're putting all of our sequences in order of least to most mutated. And we can see here classic clonal burst events. And this is an event where a single mutation is so successful that it lets the cell take over an entire germinal center. What's unusual about the gut is we see this happening again and again, indicative of ongoing selection over time. When we produce the single mutated clone as a monoclonal antibody, we could show that they bind to the microbiome, and this is in a flow cytometry-based assay. But what's really interesting is if we take away all the mutations that are required over time, we can lose binding to the microbiome. So what I'm showing you here is that mutations gained in germinal centers improve binding to the microbiome. When we use simplified models of the microbiome that only have 12 bacteria, we could show even that the heavily expanded clones could bind to one individual member of the microbiome and completely lose binding when we take those mutations away. After we saw this, we decided to ask the question of how germinal center selection in the steady state is affected by changes in the microbiome. So when we take away the microbiome altogether, and this is in germ-free animals, we were able to observe that selection still occurred even in the absence of the microbiome. And here we see this huge, dense, strongly selected germinal centers, which after 4 weeks completely turn over. I've included this cool extra piece of data actually here, where we see that if we use the less complex microbiome, oligo-MM12, we see an intermediate selection speed. So it looks like antigen diversity plays roles in selection speed, where the least diverse microbiomes, or the absence of the microbiome altogether, accelerates germinal center selection. As we looked at the sequences from germ-free animals again and again, we began to see an extraordinary phenomenon. We saw the emergence of public clonotypes that appeared again and again in different mice. Here I'm showing you just 2 of our favorite public clones using the V region 147 or 112, and these are colored in the corresponding bars. We decided to ask whether it was actually a rare occurrence to find these cells. So we compared our sequences to naive B-cell databases that were publicly available. And we can see here that whilst we see these clones in 3 or 17% of all of our sequences, this is compared to 1 in 3 million or 1 in 17,000 naive B cells. So these are hugely enriched in the repertoire of germ-free animals. To get a more comprehensive picture, we scaled up our sequencing approach and compared 3 groups of mice. SPF, that's diverse microbiomes, germ-free, no microbiome, and MM12, less diverse. When we looked for our public clone, we saw it in all germ-free animals, and surprisingly, very frequently in MM12 germinal center repertoires. When we zoom out and plot the sharing of any identical sequence, we see a picture that looks like this, and I'll just go through a couple of features of this plot for you. Firstly, one thing to note is there are clearly more than 2 public clones, and sharing is actually extensive in the germ-free environment. These are our blue bars. Each bar is an individual mouse. Each line is an individual sharing event. These sharing events are somewhat maintained in the oligo-MM12 minimal microbiome setting, where you can see lots of cross-sharing events, as well as a few inter-sharing events. These are probably to one of the 12 bacteria. Interestingly, in our SPF complex environment, we see almost no crossover events, although we do start to see the emergence of some shared clones. This is very infrequent, and these are probably towards the most abundant bacterial species. And I'll lastly just make the point that if these repertoires were truly polyreactive, we would expect equal sharing between all conditions of our data. And if the Payer's patch just generated diversity and only functioned in that capacity, we would actually probably see no sharing events. So just this piece of data alone makes us convinced that the microbiome is shaping the clonal evolution of responses in a very meaningful way. So to summarize some of this early data that I've shown you, the jackpot selection events occur in mesenteric lymph nodes and Payer's patches and contain single heavily expanded clones, which have all the hallmarks of clonal bursts. Some of these clones bound the microbiome and underwent true affinity-driven selection towards single bacterial species. Germ-free mice have germinal centers that select rapidly and contain public clonotypes. And largely, germinal center behavior and clonality is tuned by both the presence and the complexity of the microbiome. So I'll leave you with a kind of a bigger picture outlook of how the commensal antibody feedback loop works. So we know that during infections can induce germinal center formation, and within these germinal centers, clones evolve that are very specific to the antigen that is incoming, and therefore very specific antibodies are formed which bind to the infectious agent and mark it for clearance. However, in the intestine, I hope that I showed you today that some of the members of our microbiome can induce germinal center responses, which do undergo strong selection events. However, how much these germinal centers actually contribute to the secreted antibody pool is really unknown. And also what these antibodies actually do to the bacteria that they bind to is a massive open question. Others have shown that the antibodies formed in the intestine clear bacteria, whereas groups have also shown that they mark them for adherence and actually increase their numbers, whilst others have showed that antibodies change bacterial fitness and alter lifestyle choices. So this question is still really open. And ultimately, we're interested in what happens when you get an intestinal infection in the background of having these ongoing germinal center responses. So with that in mind, my lab understands the interplay of B cells and the intestinal microenvironment, largely split into these two questions. How does the intestine impact B cells? And how do B cells impact the intestine? So we're really interested in questions of timing, location, and function. And I'll leave you here with some thank yous. So I'll thank my lab and the lab of Gabriele Victoria and Dan Musida at Rockefeller University. I'll thank you all for listening and thank my funders who supported this work. And lastly, I'll end by saying a huge thank you to the Michelson and Science Prize for Immunology for highlighting this work today. Thank you very much.
Thank you, Carla. You can enter your questions for Carla in the Q&A box as well. And after our next presentation, we will open up for group questions. So finally, please welcome our grand prize winner, Dr. Bingxu Liu of the Institute for Protein Design at the University of Washington, Seattle. Bingxu studies how the innate immune system senses danger signals and enacts protective mechanisms through the STING sensor. Using a combination of genetic, biochemical, and structural tools, he discovered the human STING plays a surprising role as a proton channel, the first ever immune sensing channel to be reported in humans.
Hey everyone, thank you Shannon for the nice introduction.
His presentation is titled Protons Are the New First Responders to Danger. Human STING's newfound function as a channel expands our understanding of immunity.
In the last 2 decades, we really see the power of harnessing immune system to cure diseases. We know a lot of immune proteins have multiple downstream functions, which are known as pleiotropic effects. To get an optimal response of the immunotherapy, we really want to just get the beneficial part and avoid the harmful part. To precisely perturb the immune system, we need to clearly understand the biological mechanisms of immunoproteins. STING, for example, plays an important role in a lot of different biological processes. And we know activating STING can induce a lot of downstream processes, but we don't know how they are induced. In our work recognized by the prize, we found a surprise. STING functions as the ion channel. This helps to explain how a lot of downstream functions got induced by STING. And in the same time, really open up a lot of new hypotheses that can be tested. For example, what are the roles of the STING as ion channel function in the non-immune cells, like in the neurons? Like, can it be the bridge to sense the pathogen invasion, to send signal to our neuronal cells? And also, it gives us the opportunity to see if we can develop a better therapeutic. For example, can we drug STING to act the immune system without killing the T cells to get a better therapeutic responses? We don't know the answer yet, but this is the area of research I find is very exciting.
Hello everyone, my name is Bingxu Liu. I did my PhD in MIT Biology, co-advised by Neha Cohen and Darren Irvine. I'm currently a postdoc in David Baker Group at UW. I'm really grateful our work is recognized by Michelson Philanthropies Science Prize, and it's my honor to have a chance to share our work here in Keystone Symposia about human STING is a proton channel. So when we talk about biology, the most intriguing part is the dynamics of the dynamic evolution process. And then among all different systems, my favorite is immune system, since this fast change due to the host pathogens arms race. As simple as bacteria, you need to fight with the phages, and as human, we need to constantly deal with different pathogens ranging from viruses to parasites. And this selection pressure by the pathogens really give a strong motivation to being creative of our immune system. And among different immune system, STING is one of the classic examples for this. On the one hand, you can see the similarity between the very ancestral bacterial STING to human STING, indicated by the similarity of the ligand binding domains. On the other hand, you can also see the difference. For the bacterial STING, it has the tear domain. For the human STING, it has the transmembrane domain. And the function of the STING in this in bacteria and human are totally different. So it's intriguing for us how the human STING using the very similar ancestors to build a totally different functional platform. In order to understand this question, we really need to start from the discoveries the field have made in the last decades. So the STING agonist cyclic dinucleotide can be produced by bacteria or host cells upon sensing the double-stranded DNA. Once the agonists bind to STING, they induce a conformational change to let the STING translocate from ER to Golgi, where it start to oligomerize and induce downstream functions that including interferons, non-canonical autophages, and NLRP3 inflammasome activation. Well, we have pretty good understanding of how interferon is induced by STING activation. For the two very important pathogen defense processes, we still don't know what are the linkage between STING activation to these two process induction. What we do know is both processes are usually triggered by the pH increase upon pathogen invasion. When they break the integrity of these vesicles, the proton will come out, and then there's a pH increase to be sensed by these vesicles to initiate the two downstream functions. And since STING can induce both two processes, it's very intuitive for us to hypothesize that maybe STING also can induce a pH increase to trigger these downstream processes. And to test this hypothesis, we really need organelle-specific pH reporters. And going through literatures, we decided to use a genetically encoded pH reporter consists of two fluorescence protein, SAP, which is a green fluorescence protein that is sensitive to pH change, and mRuby that is insensitive to the pH change. By tagging the fusion protein to organelle targeting sequence, we can put this pH reporter into a Golgi. And if the Golgi maintains this acidic environment, we see the SAP being quenched and mRuby is fluorescent. And if there's a pH increase, we expect to see the SAP to be fluorescent, and while mRuby kept the same. And so in this experiment, using the cell expressing this pH reporters, one group we just treated with DMSO, the other group we had the STING agonist, the ABCI, and I hope you can appreciate that upon STING agonist stimulation, we see a dramatic increase of the SAP fluorescent signal, indicates there's a strong pH increase inside these vesicles. So this is really nice to validate our initial hypothesis STING potentially can trigger the pH increase, but it still doesn't answer how. There are different ways for STING to achieve that. The most obvious one will be STING maybe active another ion channel to achieve these processes. But from a CRISPR screening we did, it seems unlikely that's the case. Well, there's an alternative hypothesis that's much crazier, that maybe STING itself actually can serve as a channel. So this is crazy because there haven't been one that in the mammalian system that immune sensor protein itself functions as a channel. But in plants, there are actually cases that immune sensors serve as a channel to execute the downstream functions. And when we come back to really took a look at the full-length STING cryo-EM structure, there indeed a hole that's spanning through the transmembrane domain of the STING that give the possibility that this could be a channel. But then how could we block this proposed or to test our hypothesis? So we really got stuck here for a very long time until this paper published that report a structure of the STING with a non-canonical agonist CF3. For the canonical STING agonists like cGAMP or DIVZI, they bind to this ligand binding domain to initiate the activation. But for the non-canonical agonist CF3, they bind to the STING transmembrane domain. And if you look carefully, that's exactly the proposed pore area inside the STING transmembrane domain. So now the obvious question for us is, can CF3 actually block the pore function of STING? So I showed you this experiment before, but now we have additional group. In addition to a DMSO group and the STING agonist group, we now have the STING agonist plus a potential pore blocker, CF3. And I hope you can appreciate the drastic difference that with the STING agonist stimulation, we can see a strong pH increase inside of the vesicles. But now with CF3, the increase is much reduced. That suggests that CF3 is potential pore blocker and STING is a potential channel here. But this is a cell experiment. It's very promising, but it's not a golden standard to prove if a protein is a channel or not. The golden standard will be the proto-liposome experiment. So we decided to we purify the STING and put on liposome to make the proto-liposome. And the assay was run in the following manner. So we have a STING containing proto-liposome with high potassium buffer inside. And then we also have the pH-sensitive dye ACMA. And when we treat the proto-liposome with valinomycin, which is a potassium ionophore, the potassium will diffuse from inside the liposome to outside liposome, driven by the concentration gradient. And then this diffusion process gave us the voltage difference across the membrane. And if the STING is a bona fide proton channel, it will allow the proton will get into the liposome driven by the voltage difference to acidify the liposome. And the acidification process will quench the ACMA dye. And at the end of the assay, we will add the CCCP, which is a proton ionophore, to finish the reaction and shows the assay works. And if STING is indeed a proton channel, we will expect for normal empty liposome, there will be no ACMA quenching effect. And in the STING containing liposome, we will expect the ACMA dye quenching effect. And if the CF3 also can bind to STING, we will expect a reduced quenching effect. And the result is indeed consistent with what we expected. When we just have the empty liposome, we see no big difference of the the relative fluorescence upon valinomycin. But when we have the STING containing proto-liposome, that we see a pretty dramatic ACMA like quenching effect. And moreover, when we give the CF3 to the STING containing liposome, now the quenching effect is also reduced. So that's really telling us, really suggests that the STING itself is a proton channel, and CF3 blocks the STING channel function potentially by binding to the proposed pore area. So if you remember the original motivation, original hypothesis about how STING induces two downstream processes is by the pH increase. And now we have the good perturbation CF3 to block the pore area. The next question is, can CF3 also block the two downstream cell biology processes? The non-canonical autophagy process can be marked by LC3 conjugation. And here, when we just give DIVZI, we see a pretty clear conjugation. And then this conjugation can be blocked by co-treatment with CF3. Similarly, the NLRP3 activation process indicated by the the puncta formation on the vesicles here can also be blocked by co-treat with CF3. Thus, I hope this is a story that we basically start from a basic question: how STING actually induce two downstream processes, and then we find out a good hypothesis, a good theory to expand this. That STING function as a proton channel to change the pH to induce the two downstream processes. And this is what we have done here, mainly cell biology work. And the fact that this is a basic cell biology processes really open the door for us to explore further in the physiology settings. Like, for example, what STING can do in neuronal cells or T cells, where they really care about the vesicle voltage and the ion concentration in different vesicles. And then now we know STING is a channel, we can form a lot of different hypotheses to see the physiology relevance. And this is a teamwork that between the two labs I was in, Near Calhoun lab and Darwin lab. I got a tremendous support from both my mentors and also the team members, Rebecca, Ivan, Matteo, Ali, Katan, Mark, and Matt, and our external collaborator, Chris, Paul, and Songgang. And thank you all for your attention here.
Great. Thank you all for joining us today to share your inspiring work with the audience. I'll start with a question for Dr. Liu as our audience starts to enter some of their questions for you. Dr. Liu, can you tell us more about the implications of your discovery that STING acts as an ion channel, specifically in the context of neuronal cells, and what special effects it could have there?
Yeah, so that's really interesting question, and something we have been thinking a lot. And the special thing why we think a lot about immune, the STING function at the channel in neuronal cells is because the ion flow and the voltage change have been, the language that the neuronal cells like to speak, right? And then the fact that now, we have a immune sensing protein function at the channel that potentially upon sensing the agonist and then to induce the ion change and potentially voltage change. We have a lot of different hypotheses, and we think that's potentially when you face an infection and when STING is activated, and that potentially, for example, really changing if you can feel the infection like in the real time, or if you can actually, the neuronal cell potentially will secrete some hormones or other things to counterattack to regulate the whole immune system. So we don't know the answer, but every time, once we find a new cell biology discovery, and then you can build up based on all the knowledge pieces you have. And I think that's potentially a really interesting thing to ask and then to do experimental check.
Are there currently known hormones, as you mentioned, or things that neurons secrete to stimulate immune reactions or immune activation?
I think there are actually a lot of recent, I cannot really name a lot of very specific ones, but then there are a lot of recent, I think especially maybe Carla will know better in the gut. Actually, there's a lot of gut immune, the neuronal immune reaction there. And I think that's a very interesting area. And recently people also discovered that STING expressed in a lot of different neuronal cells, especially sometimes different in mouse and human. But exactly, what type and then how they may be different behave, I think that's all great open questions.
Interesting.
And I also do want to highlight, although T cells are not really, the immune cells, they're also sometimes very sensitive to the, voltage change and ion concentration inside them. So the channel function could really have interesting, a role we haven't really appreciated so far.
Interesting. Yeah, Carla, do you want to expand on anything about neuronal influence on immune activation in the gut?
Yeah, there's lots known. Very recently, there's papers on bioarchives about sensory neurons controlling B cell function through neurotransmitter release. So that's kind of, now we think of neurotransmitters as a kind of cytokine even. So that's kind of emerging. But yeah, it seems like week on week we find out more that the immune system and the nervous system interact.
Very interesting. We do have an audience question for Carla. The question is, have you studied the interaction of the B cells and microbiota in inflammatory bowel disease?
Yeah, it's a very good question. We're looking into it right now, actually. There's lots of complexities with IBD. Mouse models and human disease are not perfectly aligned. So that's one of the major issues when looking at IBD in the lab.
The other thing is the role of B cells is quite murky in IBD. So if you give humans B cell depletion therapy, their IBD gets worse. However, there is other studies that show that probably the germinal center particularly can make pathogenic antibodies that worsen disease course. So there's some B cells that are beneficial and there's some that are not beneficial, and that we don't understand. So it's not clear what we should be depleting and what we should be favoring. And yes, it's an active area of study in lots of people's labs, including mine.
Interesting. I did have something similar to ask about, do you see the same germinal center reactions and processes happening in those mouse models of IBD? Or does the whole process kind of happen differently with regards to microbiota intolerance and that sort of thing?
So stand by. We'll be letting you know soon. But what we know is that lots of the IBD pathogenesis is in the colon and not in the small intestine. So classically, Peyer's patches don't exist in the colon, but there are small structures known as cryptopatches, which probably contain something germinal center-like. So TLS, these tertiary lymphoid structures, are probably going to be very relevant during IBD, progression of IBD onto things like colorectal cancer. So whether this is all happening in the lymphoid organs or whether it's happening locally in the tissue is yet to be thoroughly established, but probably those two things are going to be interacting.
Interesting. And I have a question for Gabriele. Really exciting research and results you've seen in mice. What would the next step be, and what are your next plans to get to test this approach in humans with AML?
So yeah, we are currently working on that. We're trying to scale up the process to develop as a clinical grade process for the manipulation of hematopoietic stem cells for allogeneic bone marrow transplantation. A very similar approach is already being conducted as a clinical trial by VOR and also at UPenn, which have designed an analogous strategy with the modification of CD33 as a knockout, not as epitope editing. So this is currently going on. So we are trying to replicate sort of a similar kind of trial, but probably with the complexities with additional epitope editing using base editor and so on. So we are the first of this kind for us. So we are currently working on that. And the other point is that I'm discussing with the physicians what would be the best first clinical trial, because it's very being related to an allogeneic stem cell transplantation and potentially with also the use of CAR T cells after allotransplant. It's quite a complex therapeutic approach, but we think we can make it work. So this is currently going on, and we hope to get to an IND application by next year and a half.
Oh, great. So if you get an IND by next year and a half, when would you first treat a patient, you think, as a test? Is that right after that? I'm unfamiliar with the timeline so much.
No, we'll probably be in the subsequent, again, year or so. But again, I cannot give any specifics about the timeline as of now. So we'll see. I'll try to keep you posted about the clinical development of the epitope editing.
Oh, that's very exciting. And could you kind of envision expanding this approach beyond hematologic malignancies to other cancers? Would that work? Or what would the hurdles be?
That's actually, there are, I think, very difficult to overcome limitations in that sense, because so you can imagine that you can modify any human tissue to avoid off-target toxicity of immune therapies or even other therapies like small molecule inhibitors. But the problem would be how accessible or is that organ to be modified, or how good is your delivery vehicle and in vivo targeting system to modify that. And I think it makes more sense in non-malignant conditions, because again, if you shift the manufacturing, so the gene engineering from ex vivo to in vivo, you need to be very careful with the targeting, because you always risk modification of the cancer cells as well. So that's why we started off with hematological cancer. The healthy counterpart is easy to access and modify ex vivo. And again, it works very well even in an allogeneic setting. It's going to be more complicated for other organs, but probably for other types of therapies or non-neoplastic diseases, it might be a feasible approach.
Okay. And then are there other hematologic diseases that are not malignancies that kind of alternatively you could use the approach for?
Yes, actually, we've been working with other antigens as well. So potentially you can apply this to any other antigen as long as you validate the mutation that you're inserting in the extracellular domain. If we are talking about immune therapies, there's a whole field about modifying the hematopoietic stem cells, not therapy, but to reduce on target of tumor toxicity of small molecules inhibitors, for instance, and so on. So that's a super hot topic as well recently. But finally, I think the other very straightforward approach of the epitope editing strategy is in bone marrow transplantation, because we are used to exploit chemotherapy or radiotherapy as pre-transplant conditioning to make space in the bone marrow niche. And that obviously are very toxic, and they produce a number of side effects that are even long-term or even irreversible. And so one way to avoid that is to use non-genotoxic approaches, and again, using immunotherapies. But again, on the other end, it's difficult to use them because of if you use antibodies, you have a limitation related to their pharmacokinetics. And so that's another very interesting setting where you could imagine modifying the donor cells or the autologous cells if it's an auto transplant, and make them resistant to the antibody conditioning. So you are free to choose however you want to administer your antibody. If you want to give them everything before the transplant, very close to the transplantation itself, or even afterwards, to achieve in vivo selection. And again, in vivo selection would be another super interesting approach for gene therapies, especially if you imagine, again, non-neoplastic diseases where you can modify stem cells in vivo. This is going to work very well by in vivo delivery, because you can introduce your therapeutic gene editing together with an epitope modification and then select for those cells in vivo using an antibody or an ADC, something like that. So this is something that we are currently working on to demonstrate as a proof of concept.
Very interesting. Sounds like a super versatile approach. Exciting to see where it leads. I'll go to a question for Dr. Liu now. So you mentioned that the discovery can help to improve immunotherapy approaches. Can you expand on that more? And for which therapies and diseases in particular would this knowledge help us?
Yeah, so that's a really interesting question. And then I can give you one example for that. For example, STING have been a really interesting target people have been trying for immunotherapy for a long time. And the general idea will be if you can activate the innate immune system, that really can be helpful to really expand your adaptive immune system to tackle cancer. But one common problem people have been reported is once you use the STING agonist and inject into the mice, and you will see the activity T cell actually got depleted during that process. And that had been a mystery in the field, and the people just have no idea why. And the previous report also had been shown that T cell potentially got killed by STING agonists by some iron imbalance, especially the calcium. Well, this is not directly related to, it's kind of like a mystery how exactly a potential iron imbalance induced by STING actually can kill the T cells. But imagine if this is actually ion leakage related to the channel opening or not. And when we activate the STING, which we know the interferon signal is very important for this process, but maybe we don't want the ion leakage process in the process that deplete our activated T cells. Then we got the immune therapy we want rather than a natural, overactivation kill the T cells, right? So it's like I would really like to use the example when people start engineering all the cytokines. Is the natural immune response a lot of times are not perfect for our purpose. And if you understand, all the details and mechanistic understanding the whole process, we can take the part we want and then remove the part like we don't want. Yeah.
Great. So you can tune the response that you want. Great.
Hopefully, yeah.
That's the goal. Excellent. We have another question from the audience for Carla. Carla, is it known if prions can stimulate germinal centers in the intestines? Do you know anything about that?
I don't know. Yeah, I actually don't know. Yeah, not sure. I'll look into it.
Do you know outside of the intestine if in other
Outside of the
tissues?
intestine, I haven't heard much about prions and B cells. But
Interesting.
this is not an interaction we're paying a lot of close attention to. So it may very well be known. I don't know.
Got it. I have another question for you as well, which
Okay.
you mentioned in your presentation that we don't really know much about what the antibodies are produced doing in order to kind of make the microbiome stabilized. Can you expand a little bit more on what they might be doing, what the kind of thoughts are on these processes?
Yeah, well, I guess the traditional way that we think of antibodies is that antibodies bind to something and clear it. So that has been shown in the gut towards microbiome, for sure. But also it's been shown that antibodies can bind to bacteria and make them more adherent to the mucus layer of the gut, therefore actually targeting them for adherence so that they remain. If you think about that, it's actually quite difficult to understand how you would ever stop producing antibody, because you produce antibody, it sticks, the bug binds, there's more bug, you make more. So that's kind of a difficult thing to conceptually understand. And then other groups have done things like giving the same bacteria again and again, and then pulling out the antibodies and seeing what they do. And almost every individual antibody does a different thing to the bacteria. So some can change the metabolism, some can change how these bacteria move, so how their flagellin functions. Some can change how sensitive they are to metabolites in the environment. Some can simply clear. All these kinds of different outcomes, sometimes to the same bacteria. So we're not sure whether it's a feature of the bacteria or the antigen that can drive this sort of thing, or a feature of the context where this antibody is seen. If it's seen in the context of another signal, these are still things we're trying to work out.
Very interesting.
Yeah.
And do you think similar processes are at play, both in terms of the germinal centers as well as what you just mentioned with the antibodies, in other places around the body where there is a microbiota, like the skin, the mouth, other kind of,
Oh, undoubtedly. Yeah, undoubtedly. However, the lung seems to be a bit more quiet in terms of steady state germinal centers. These probably form over long time periods during aging. The mouth, very, very likely that these are forming somewhere specifically in the tonsils, of course. But yeah, very likely.
And are you and your team looking into that, or other scientists looking at that?
We're interested in mostly gut draining. Skin is kind of a complicated situation. We don't see much in steady state skin, germinal center wise, but in lots of high or outside of normal inflammation settings, you do see germinal center formation right in the site of infection in the skin. Other groups are looking into this, yeah.
Great, thank you. And now I'll ask a general question to all of you, and you guys can have a group discussion or you can each answer individually, but what is the next step in your research, and how will the Michelson Science Prize help you to advance your work that you would not have been able to do might not have been able to do without the prize? Maybe Gabriele, maybe you can start for us.
I think the Michelson Prize and Science Prize for Immunology has been a fantastic occasion to showcase the work, really. I think it has really created a lot of attention to all three of our work. And also the fact that we are having this discussion and this fantastic online panel, I think it's already a recognition of that. And I think I was able to connect with people. I think we have created a lot of discussion that people have reached out and ask about the project and how that can be employed and so on. So I think there's a perfect way to spark new collaboration with other scientists and simply get beyond publication the work known on different platforms.
Yeah, and I think for junior PIs, we're always looking to attract talent to come and work with us. So having rising PhD candidates or postdoc candidates reading the work and then connecting with us in order to discuss it and come and potentially work on something that they're interested in is really useful for us as we're establishing on our own.
And I think I'm really grateful because this is actually a really good way. First, it's a big recognition of our teamwork, as I showed in the acknowledge. It's really a teamwork, so everyone got really happy that our work got recognized. And second, as I think Gabriele and Carla have mentioned, that after the prize, I think we actually got reached out by people from different fields that from a lot of angles we even haven't thought about, because this is more of a basic research, and then that's really inspired a lot of new collaboration. The thing is, I left the lab 1 year ago because as a graduate student, I move on to my next stage. And then I think the past work, basically, we can discover how nature programmed the immune system. And now I moved to my postdoc stage at the Institute of Protein Design. We're trying to say, okay, if nature haven't reinvented this, can we invent something to really do the job? And I think that's really deeply inspiring. And a lot of these new directions actually come from discussion with people who after they recognize our work from the Michelson Foundation Prize. And also I think one more point I want to highlight as an international student, it's the first time for me to write non-manuscript, well, how to say that? This is more of a public-oriented the articles, and the writing process itself, I had so much fun, and then discussing with people, and then I think it's really good training for my scientific communication.
Interesting. Yeah, and I think that's kind of a common theme we've heard from a lot of the Michelson awardees over the years is not just the visibility, but the conversations it stimulates and where those lead in the unexpected directions. Do you guys have any more to expand on kind of unexpected directions that have already come out of this for you?
I just think I agree. I think it's really important to focus on engagement with people who wouldn't be very familiar with your work otherwise. So writing for an accessible audience is something we don't get to do that much if we're writing grants and papers. So it's really enjoyable to be able to like frame your work in a way that most scientists or even most people who are not scientists could understand. So that's been like very valuable for me.
I think that's definitely a skill that some scientists have innately, but I feel like a lot of us need to work on. So I think it's great. And I don't know, for me, when I'm doing things like that, it makes me realize the kind of broader impacts when you're kind of stuck in the details in the lab, kind of to open up and really look at the breadth of these impacts in other fields that you might be touching. Great. Let's see, we have another question for Dr. Liu online from our audience. And yes, we've got a few more minutes left. So audience, please submit your questions if you want them to get answered. Dr. Liu, are there other STING-like proteins or proteins with sequence similarity to STING TM domain? And how different is STING channel with other channels, and why hadn't it been predicted by sequence similarity or structural similarity?
Yeah, so in the beginning, like first, that's a really good question. Basically, like the simple answer is we haven't really done well. We did like very simple sequence analysis when we just started this project, and it's not the classic, it's not the classic, for example, the potassium selection channel motif. And then we also want to say is like, although we know STING can transport protons, we actually are not sure if STING can only transport, protons. We don't know if STING can only transport protons. Maybe there are other, like ions can also be transported. So that makes it pretty hard. It's kind of, we don't really know where to align to, but at least, funny part is our lab is not even a channel lab. We did this purely by speculation of this may have this function, and by collaboration, and then to basically figure out. And then so the simple idea is maybe there are actually a family of the channel protein that's very close to STING motif, and maybe it's a new motif, or maybe for a more promiscuous channel that there's no like a very fixed motif for this. But I really like this question. I think this could be really interesting if we people can actually taking the channel motif of STING and do alignment to other potential proteins, either in mammalian world or maybe in bacteriophage, to see if there's any similarity. And more interestingly, as we said, this is the evolution process to gain this process, how step by step, and then the channel function actually got acquired is also quite an open question, and I think it's really interesting to be answered.
Oh, really interesting. Yeah. And I have a related question. Having discovered such a novel function for a Sting sensor, was it hard to convince people or even yourself when you discovered it that this indeed was happening as this novel mechanism?
Yeah, we initially have a theory about 2019. Actually, it was like basically I think my previous project got scooped, so I need to I need to keep reading other things, and I just like reading papers. And after reading, there are several really inspiring papers I mentioned in my slides. As for example, the 2019 Feng Shao paper discovered a lot of this like a bacteria invasion process to actually induce non-canonical autophagy are actually by the proton leakage and other interesting rare area, like make me connect the dots. But as I mentioned, we're not the channel lab, like like we just haven't worked on this. It's just a theory, and then it's really hard for us to test anything. It's really, I think, happened during 2022 when we saw the paper from UT Southwestern that they found that CP3 actually bound to the Sting in the cryo-EM structure. We're like, maybe the theory is not too crazy. And then it's very easy for us to buy a compound to try to see if we can separate the Sting activation versus Sting activation of the interferon versus Sting induced non-canonical autophagy. And the compound is like $50. So basically, we just buy the compound that we have a cell line already, and we have also, and after that, we're like, okay, this is pretty clear, what's going on. It's very likely to be a channel. And then, but we still unsure. I think, talking with people, the Sting expert from, UT Southwestern and the channel expert, like Chris Miller from Brandeis, and then also the Sting in-house Sting expert like Rebecca and Matteo inside our lab. And we start to see, okay, maybe it's actually now, since we reached the first milestone, and then we can, try to put more effort into it and actually finally the project. So there's the big gap. We have the theory, but then we're not even, brief enough to put a lot of resources to test it. And then the final stage, we have all the things ready and then quickly test it out.
Interesting. Very exciting work. So now I want to kind of open it up to you guys. Do you have any questions for each other, having come from different kind of parts of the immunology field, but obviously experts in everything that you do, or any insights or thoughts you have about each other's work and questions? Yeah.
I have one for Bingxu. So Sting, I know, is involved. It's not my field, but I know it's involved in a number of autoinflammatory phenomena, inheritable diseases, some with vasculopathy and other features that are both immune and hematological and non-hematological. And I was wondering if the knowledge, so the realization that Sting is a channel, does that change the interpretation, or does it add something on what we know about the pathogenetic process in these inheritable diseases? Have you looked into that, or is there any other, maybe potential explanation for some of the clinical features of these diseases?
Right. I think that's a very excellent question, and I think so. There are two things. One thing is kind of, so for example, some of this effect can be explained by Sting activation causing the interferon secretion. And I think that part is probably independent of the channel function, channel part of the function. But I think there's a lot of other things like people are known that's potentially not related to the, the interferon. For example, the T cell death that I mentioned, right? And then that will be very interesting to see if the channel function really do anything to this pathology process. One thing I think the field moving on could potentially do, because right now the only perturbation we have to separate the interferon part versus the channel part is by the CC3 molecule, which by itself also a Sting agonist for interferon. So moving on, I think if people actually can make the Sting mutants that only have one part of function but not the other, I think that will really be helpful to answer exactly the question you ask, and then that will be really exciting, basically to know if how much of the role of the channel part play and how much the role of the interferon play during all these processes.
Thanks.
I actually have two questions, I think, for each of the authorities. Yeah, and I think for the first question, I think it's really beautiful work for the Gabriele, basically to try to basically remove the own antigen epitope and then be targeted. And then I was wondering, like it's probably people already tried this. It's like, have people just tried to put an inhibitory, like, receptors into the the HSC you want to protect, or what's the difference of just editing away the epitope, or just that your CAR T still can recognize, but then it cannot kill?
So if I understood correctly, I mean, what about if we instead express an inhibitory molecule that is recognized by the CAR, like as a inhibitory negative CAR, that would prevent the activation of CAR T cells? So we reasoned about it. We were thinking about testing that approach, and we had some preliminary data that obviously feasibility in theory is possible in that sense, but on the other end, my idea was that it's much more elegant and a cleaner approach if we just remove the on-target recognition at all, and we avoid the expression of a second transgene in stem cells and therefore also the entire progeny. So my impression was I didn't run a comparison between these two approaches, but the idea was I liked best the idea of avoiding completely the on-target activation of tumor, of CAR T cells or whatever other immunotherapy you want to use, as if the antigen never existed in the first place, as compared to have a complex circuitry within the T cells, which you also need to titrate very well, how the activation part is blocked by the inhibitory signal. And I think that makes for a more complicated and maybe incomplete elimination of the target of tumor toxicity. So overall, because again, as soon as I realized that it was possible to introduce a single epitope mutation that were enough to abrogate most of the binding affinity, and that this mutation could be made safe enough, it's because some of these are even present in other mammalian or even non-human primates, so they are preserved in terms of functionality. I thought that was simply the easiest and cleaner approach. Otherwise, you would have need to again transduce probably your HSCs and so on with another lentivirus, which again everything is feasible in the realm of gene therapy, but I think this way probably works slightly better. The advantage on what you just mentioned, this proposition, is that it's agnostic on the antigen part. So any CAR would be made ineffective against the edited hematopoiesis because you don't need to edit each single individual antigen.
Right.
But we were looking into that. Actually, we are some experiments running for a proof of concept, but it's much of early stages as compared to the epitope editing story.
Yeah, I like the angle you say. Basically, even for delivery, it will be much easier because you can potentially do in vivo just editing the natural one. Yeah. And I have a question for Carla also, because this is actually quite interesting. It's like, when you are talking about all these different things that actually, in the germ-free mice or in the, oligo colony mice or in the just like much more complex, microbiome environment with the wild-type mice, it's like, a natural trajectory of, for example, human or mice taking the antibiotics basically mimic the whole steps, right?
Right.
You basically will start have a germ-free mice and then probably some oligo, just several bacterial strains, and you have. And then, do you see how would you imagine the germinal center response would dynamically change during the restoration after the antibiotic treatment?
Yeah, good question. So we've done this in several ways. So if you just give antibiotics, you can pretty much get rid of almost everything. But if the GC niche was already established, it's quite hard to change it. So you don't reset it as much as if you were germ-free the whole time. However, if you give germ-free mice and the bugs, and you just colonize them in adulthood, they do seem to restore pretty much to what we would expect in SPF. So there's something to do with establishment of the niche that's important, and probably this is to do with the weaning reaction. So probably there's a period of time in early life where the immune system, we know this very well for T cells actually, but probably in B cells as well, there's a period of time whereby you're programmed, and then it's kind of difficult to reprogram. So this is very therapeutically relevant in situations where children or pregnant mothers are given very high dose antibiotics.
Right.
So that's kind of where we're looking now to see if the time window of colonization causes long-term repertoire changes, and if these repertoire changes are actually the thing that predispose you to chronic autoimmunity.
That's nice. Yeah. And how much you imagine, like, because when you do all these reactions, you feel like they're actually in different mice, there are some shared epitopes, right?
Yeah,
like, but
correct.
will you imagine, like, I just wonder how much, or maybe you already done experiment, like in the, antibiotics treatment and restoration steps, how variable you will imagine that, or maybe there are some bacteria just recover much faster. So you always kind of follow the same trend.
Yeah, it's a good question. We haven't checked super thoroughly, but it's important to mention that these public clones don't bind bacteria.
Oh, I see.
Because they come from germ-free animals. So we checked if they bound bacteria, no. Self, no. Polyreactivity, no. So we actually can't figure out what they bind to. We also can't even figure out whether they ever make antibody. Because germ-free mice have this other interesting phenomenon where they're full of germinal centers, but they have no antibody-producing plasma cells in their gut. So there's some sort of dead end that happened in these germinal centers that don't have bacterial signals. So that's kind of interesting. We don't know what the signal is that makes you leave a germinal center and go to the intestine. But we do know that germ-free mice can't make IgA and don't have any IgA class switching, and that these public clones are always IgG1.
Interesting. So it's time to wrap up our discussion now. Thank you all so much. It's been a really fascinating, engaging discussion. I really enjoyed these questions at the end. Thank you all, our audience, for joining us today to discuss these advances in innate and adaptive immunology and immunotherapy research. The ePanel will be available on demand, and we will email all the registrants when the recording is available. If you did not get your question answered, please join us in the networking lounge. And even if you don't have any further questions, we'd love to see you in the networking lounge to continue these conversations and make connections. And the Remo platform allows you to jump between small group discussions as you would during a coffee break or a cocktail hour. So you can meet with the panelists, you can meet fellow audience members and see what they're working on and make those collaborations and new insights that we were just talking about in the discussion. So click the button to join on the homepage or on the closing screen once we conclude. Finally, scholarship and abstract deadlines are approaching for upcoming Keystone Symposia conferences on innate immunity, B cells, and cancer immunotherapy. See the bottom of the event homepage for more information, or visit the Keystone Symposia conference page to see all upcoming immunology conferences. Thanks again for joining us today, and we hope to see you in the networking lounge.